Good morning, everyone. My name is Camilo Paredes Tomas. My partners are Maritza and Jorge Luis, and we work at the Research Institute of Tropical Fruit Crops in Cuba. First of all, we want to say thank you to the Tropicsafe project to get the opportunity to be here. And we're going to talk about trial transmission of Candidatus Liberibacter asiaticus by Diaphorina citri to citrus plants in Cuba. In our country, Diaphorina citri was detected early since 1999. However, the first symptomatic plants were detected 7 years ago. This is in 2006, and previous worker informed Candidatus Liberibacter asiaticus as the unique species of Liberibacter associated with HLB occurrence in our country. During the Tropicsafe project, the observations support a broad dissemination of both the pathogen and the vector in all citrus areas in our region. When you screening for insect vectors, PCR assays of field-collected insects may provide indication of a possible role of a given species transmitting plant pathogen. However, as you know, some pathogen may be acquired but not injected when feeding. And for this reason, the transmission test is the final evidence needed to confirm this fact. In Cuba, there is a lot of experience that show the association with the present and the population level of Diaphorina citri with the presence of the temporal progress of the disease on the field. However, transmission tests have not been performed yet, and this is why there is a lack of conclusive and direct evidence to this conclusion. For this reason, in the framework of the Tropicsafe project, we performed several transmission assays to verify under all conditions the class transmission to healthy citrus plants through Diaphorina citri. What do we use? Well, as plant material, we use healthy seedlings obtained through seeds grown under green conditions. The sanitary status of this healthy was confirmed for the absence of the bacteria using a nested PCR with specific primers to the bacteria. And as insects, we use individual field collected in the affected area, mainly on symptomatic trees. This is plants that show symptoms in the field. What do we do? Well, we perform 2 assays, 1, 2. In each assay, we use 3 seedlings of Citrus aurantium, and we put 10 to 15 insects per plant, and this system was caged, was put on greenhouse conditions, and after that, the insects were individually recovered per plant and parasites, and the plants were maintained on their greenhouse for periodic monitoring to check the symptom appearance. The DNA extraction were performed using the setup method. As start point of plant, we use midrips, and the insects were extracted from pool of 10 to 15 insects. The PCRs performed were the nested PCR were established in our lab using this primer specific to class, and the purification of the amplicons obtained was performed using this kit of Roche, and the amplicons were direct sequentially in both things. The sequence was after that used for sequence and phylogenetic analysis using the BLAST and MEGA software. What was the result obtained? Well, during the periodic monitoring, all the plants in the 2 assays showed symptoms. This is mainly blotchy mottles and foliar yellowing, as you can see in the pictures. These observations are agreed as in time, and the symptoms expressed with the reporter from other studies in other countries. This was confirmed by amplification in all plants and all the pool insects used in the transmission using the PCR analysis. These results support the fact that a high percent of infected individual is important to achieve high transmission tests, as all the plants were inoculated with the bacteria. 2 pairs of amplicons, 1 per ascites, were selected for the subsequent analysis. This is for sequence was purified, was sequenced directly, and was used for the analysis on phylogenetic and sequence. What was the observation? Well, the 4 sequences were 100% similarity to each other, and also the consensus sequence showed a high percent of identity in the blast analysis with strains of class previously reported in the database. This confirmed the identity of the obtained sequence as belonged to a Candidatus Liberibacter asiaticus strain. Also, the sequence was used with another reference strain for phylogenetic analysis that illustrate that our isolate was nearly related with Candidatus Liberibacter asiaticus informed previously in Brazil. A high value of bootstrap, this is 100, supported the reliability of this branch. This close relation of the Cuban isolate with a Brazilian strain was also supported in previously work, as informed by Luis et al. In 2009 in our conditions. And all these results together confirmed for the first time the role of Diaphorina citri as vector of Candidatus Liberibacter asiaticus in Cuba. As recommendation, we like to recommend the support of these results using maybe some polymorphic region of the Candidatus Liberibacter genome. This is profile region and number of tandem repeat, as they are more variability genetically as respect to the 16SR that is more conservative. Thank you for your attention and have a nice day.